Okay, hi. So next question is from Tim, and Tim is asking about this thing, which I hadn't heard about this particular version of it, but I had heard about some that are similar. So Lucira, maybe I'm not sure how we say that. Check it, COVID-19 test kit. It says it's the only FDA approved home test kit, which may mean that they will accept the results from this, although I'm not sure that that's actually the case. And all of the test kits are at least EUA approved by the FDA, like approved under an emergency use thing. So this may be fully approved. I'm not sure. Anyway, what is this? How does this work? Okay, fine. So this is really cool. It's a little tiny machine that is kind of like your own little miniature PCR block. So the normal molecular test works by a thing called PCR, where you will have an enzyme that will first turn, first you'll kind of open up the virus. You just kind of mix it up with a little bit of soapy stuff. And then you will turn the virus RNA into a piece of DNA, which can be done. You just basically copy it, and, you know how copying works in DNA. And then you just start making copies of that DNA over and over and over again. This is a special version, which is called LAMP. And so this one uses, normally you would use two little pieces of DNA that will stick to either end, and they kind of bracket the thing that you want to look for. And so you'll take the region just before, the middle of the spike and the region just after the middle of the spike and copy everything in between. And if you're holding on to this end, like if you're holding on to the nose of a shark and you're holding on to the tail of the shark, chances are the thing in the middle is the middle of a shark. It's basically that principle. Yeah. And so you copy that, and then you can even sequence it. You can read out the stuff in the middle. If it exists, it's almost certainly the correct thing, but you can read it and confirm for sure that this is a piece of the SARS-CoV-2 that you pulled out. So this is a little machine. It takes batteries, yeah, and it's going to run one of these LAMP tests. Now, LAMP is a thing that we have tried. I got to say our success was mixed. On samples that were particularly high, generally the LAMP worked well. We were using a different version of this. So LAMP differs from PCR in that you've got a different enzyme and your primers are a little different. Like the little things you use to hold on to the DNA are built a little bit differently. They fold up, and it's kind of neat science, but it's kind of complicated to go into, and for most people they just say, I don't know, just tell me if it works, right? So let's do that version. Yeah, if you need more, whatever, we can do a LAMP explainer. But it works, and so it will search for and copy pieces of the actual virus genome, if that genome is there. And if you have the genome inside you, you are infected, or you are at least exposed. Theoretically, you could be in a room. You could be breathing in tons of it, get it all coated into your nose. Miraculously, maybe you don't get infected, but then you swab around that, pull it out, and come up as a low positive and say, oh, well, see, these tests are terrible. No, it just means in that case you had a near miss, and oh my goodness, yeah, you goofball, wear a mask. Yeah, this is it's basically doing something like a PCR test. It's pretty good. In our hands, we were doing indirect version. So we would do the LAMP, but instead of running a gel or something, because with a regular PCR, you make a lot of copies of the virus, which are, you should know the exact size, because you put one little clamp here, one little clamp here, you know how much space is in between, and so you're going to copy that little piece out. And so you should have a whole big stack of pieces of exactly that size. And you can do a thing that only takes maybe half an hour. You can run them down what they call a gel, and it just sorts out DNA by size. And you'll have a whole bunch of DNA at exactly that size. And that tells you, yeah, we have a positive. With LAMP, you get all kinds of different sizes. It's kind of crazy, but you get an accumulation. And so there are a couple ways you can read it out. You can have a fluorescent dye in there that will bind to double-stranded DNA. And you start out with no double-stranded DNA, but as you make DNA out of RNA and then more DNA out of DNA, you start to build it up. And it doesn't matter what shape it is, doesn't matter what size it is, it'll just keep glowing. The more DNA you make, the more it'll glow. And so you could just read that out. That would be sort of expensive to read. And so I'm betting they don't do it this way, and I haven't dug into the contents enough, which are down here. I'm not even sure they would say it, maybe proprietary. The other way that people do it, though, is with a pH change. So as the reaction is running, you are consuming things. You're consuming ATP. If you remember back to the currency, the energy currency that cells use, these enzymes use it too. And so you'd be breaking that down. You'd be spitting out phosphates. So you'd have some extra negative charge, I guess, floating around in there. You can change the pH eventually once you've done this enough. And if they put in a dye, and I see a nice purple tube there, and I'm wondering if that's a dye, like a pH-sensitive dye, then that dye will change colors if you have basically copied enough DNA and used up enough ATP or whatever else they put in there to make it accelerate the pH change. And so in our hands, it mostly worked, but there were a couple of cases where with LAMP, it would say one thing and then you couldn't actually confirm it. And then with PCR, it would say something else and you could confirm it. And so based on that, the PCR was much more reliable. And I believe it was, I think it was a little more sensitive, the PCR, but it's pretty close. So this is way better than a rapid antigen test in terms of being able to give you the correct answer. Much more likely to find a low infection will actually come back positive with a test like this. The low infections almost never come back positive with rapid antigen tests because they are fast, but they are the opposite of sensitive. So yeah, it's a little machine, takes batteries, you swab it around your nose, just like that, just like everybody likes. Oh, hate that feeling. Yeah. And you, there you go. You put it into the little thing, swirl it around. There you go. Stir the witch's cauldron 15 times. Snap the cap shut. The little ready light comes on, and it's going to operate. And it's probably got a little tiny heater block in here. And so normally in this process, you would incubate at a certain temperature, and then this process will just go and go and go. And it's pretty quick. Test should be ready within 30 minutes. That's normal for LAMP. It is faster than PCR. It's just not quite as reliable in fringe cases. So if I've got a choice between running PCR in the lab or a LAMP, I'm going to go PCR every time because I know that is the best possible quality of test that I can run, and it gives me the best chance of getting an accurate result. If I have a choice between rapid antigen and this thing, I'm going this thing every time, or something like this. I believe there are several kits like this out there. Yeah, and it's fine. Yeah, so there you go. I don't know. Don't often give positive reviews to any of these things, but yeah. And they've actually put in some of their benchmarking data to show that when they tested it, their positives come up positive and their negatives come up negative, and good for them. Everybody has to do this to get a test approved. They have to show it somewhere. Generally, it's buried on an FDA website, but it's nice when they actually put it in the documentation of the kit, because then you know, oh, okay, yeah, this thing was done, and yeah, it's good. It's reassuring, yeah, for people like me. So hope that answers your question. Yeah, looks pretty good. Thanks very much. This has been Ask Dr. Ben.